Hi everyone. Welcome to our video class today. As I was worried about the background noise where I was planning to be zooming from, instead we're just going to have a recorded video for class today. Please feel free to send me an email if you have questions, or also set up a Zoom meeting to talk through this material. Remember that you have an inquisitive assignment due Friday by 5 p.m. As I mentioned, I had given you an extension on the deadline in my other email. Remember that this week during lab time, I recommend that you are working on your lab report or studying for the exam. And remember that we'll have class on Zoom on Friday at the same Zoom link as before, which is also included in my email. Before Friday's class, please be sure to respond to the doodle poll about the review session timing uh, so that we can know when the best time for a review session would be, and I will announce the winning time on Friday. Um, Remember that the exam is on November 2nd, so Wednesday of next week. I also apologize for any random background noise that you may hear during this lecture, as my cat has decided it is super playtime. We talked so far about a lot of details about T cell activation in the periphery. So recall that we've seen some of the specific details of T cell receptor signaling, otherwise known as signal 1. So we were sort of focused right here on our T cells and the activation in the periphery. We talked about how that signal 1 can lead to the production of IL-2 that can signal back to the cell in paracrine or in autocrine to help be a growth factor and support the clonal expansion of our T cells. We also talked last time about the importance of signal 2 in properly activating a T cell, and we saw a little bit about how that can be involved in peripheral tolerance. And then we also talked through the general details of differentiation and effector functions that are mediated by CD8 positive T cells. Today, we're going to talk about the other side of this image, which is an understanding of the details of activation, differentiation, and effector function of CD4 positive T cells. When we think about the activation of CD4 positive T cells, of course, I want you to remember that T cell receptor signaling, signal 1 and signal 2, are really important, as we talked about last time. But in addition, we are going to be spending a lot of time thinking about cytokines, and the cytokines that are coming from the antigen presenting cell when our T cells are getting activated is particularly important here. If we were to talk about CD4 T cells overall, the big thing that we really think about with CD4 T cells in terms of their effector function is not the killing of target cells. It is the activation and interaction with other cell types, and that all happens through CD4 T cells secreting cytokines. You can see the specific details of this here, and realistically, the secretion of cytokines by CD4 T cells, when CD4 T cells are differentiated and become effector cells, we often refer to them as helper T cells. You can see this T sub H, as opposed to our CD8 cells, which went from being naive CD8 cells to CTLs. The CD4s go from naive CD4 cells to helper T cells. And our helper T cells are specialized to secrete cytokines. Remember that our CTLs were specialized to secrete lytic granules. So just like our CD8 T cell had some lytic granules in the cell, the CD4 T cell has some vesicles that contain a lot of cytokine. And so when that helper T cell interacts with some other cell type, that cell will do some rearrangement inside to secrete cytokines directionally towards some other cell. Again, because we have our integrins kind of making a wall, we can only see the secretion going directly to this one other contact cell. So it's a very directional secretion. It actually looks, again, very similar to the secretion of the lytic granules. However, the specific cytokine that is being secreted, so we're thinking right here, the specific cytokine that's being secreted by that helper T cell can differ in a number of different situations. There isn't just one kind of helper T cell. There are actually multiple different kinds of helper T cells. And the specific cytokine that the helper T cell is secreting will depend on which type of helper T cell it is. When we think about how this sort of cell decides which type of helper T cell it is going to become, it turns out that this is actually based on what happens at sort of this initial antigen recognition and activation step. So right here, our T cell starts out as a naive CD4 cell, sometimes referred to as a Th0 cell. And when it does its clonal expansion and differentiation, it will become one of our different types of helper T cells because of how this process works. This is all based on the cytokine that the helper T cell receives when it is being activated from that antigen presenting cell. So specifically, the way that our helper T cell is activated and chooses to become a particular type of helper T cell is based on the cytokine it's receiving from its antigen presenting cell. And of course, the antigen presenting cell, the innate immune cell, is producing a particular type of cytokine based on which PRRs it saw, which PRR was stimulated, which type of microbe that it saw. So sometimes people talk about CD4 T cells or helper T cells as being quarterbacks of the immune system. As you'll see, they're going to direct other immune responses in a number of different ways. If that's the case, then the antigen presenting cell with its PRR is almost like the coach that's telling the quarterback which direction it ought to go. Basically, our innate immune cell has a PRR. It's sensing microbes. That's leading to transcription of a particular cytokine so that T cells that are being activated by that innate immune cell can become a particular type of helper T cell. And in fact, the type of helper T cell that results will be particularly useful against the type of microorganism that stimulated the PRR in the first place. There are lots of different types of helper T cells that are discussed. And one of the ways that we can think about those helper T cells is that there are certain criteria that we have that allow us to say, okay, this is like a legit, real helper T cell subset. This is not some helper T cell subset that responds to fungi in your pinky on Tuesday, like I've sort of joked about. So when we talk about helper T cell subset polarization, there are three big things. You will kind of see four as we go forward, but really three big things that we have to know. So in order for something to really get to qualify as a helper T cell subset, like we're going to be talking about, first we need to know which cytokine that T cell receives, usually from its antigen presenting cell, though as you see here, it could be from some other source of cytokine, that tells that cell how to go from being a Th0 cell to whatever T helper subset it is going to be. And we refer to that as the polarizing cytokine. When that polarizing cytokine signals to our naive T cell, it gives the cell certain types of signals and allows the cell to turn on certain types of responses. One of those responses is that a certain master gene regulator, that's just a fancy way of saying a special transcription factor or a special unique transcription factor, gets turned on. Each helper T cell subset that we have has its own transcription factor that is characteristic of it. So once that cell receives polarizing cytokines, that turns on our transcription factor of interest. When that transcription factor is turned on, that allows the cell to start transcribing effector cytokines, which are the unique cytokines that are produced by that subset of cells. And so when we think about our helper T cell subsets, for each, you want to make sure that you can name the polarizing cytokine, the transcription factor or master gene regulator, and the effector cytokines. Then you'll also see that each of those different effector cytokines has a particularly important role in some types of immune responses. And as I mentioned before, the polarizing cytokines that are helping our different helper T cells differentiate into their different directions are all coming from individual innate immune cells. And so this is all sort of a big aspect of innate control of adaptive responses. This is a figure from a different textbook, and it shows you sort of this same idea where initially our antigen presenting cell will interact with a naive CD4 T cell or a Th0 cell, and it will produce some kind of polarizing cytokine. You can see some examples of polarizing cytokines here. That will turn on a particular transcription factor to push that cell to grow up and differentiate and become a particular type of helper T cell. And those helper T cell types are listed here. The definition of those helper T cell types is in fact that they have that particular transcription factor active, as well as that they are producing a particular effector cytokine. And those effector cytokines have particular roles in immune responses. When they go awry, they also can have particular roles in different disease states. I like that this figure sort of shows you going from a Th0 cell to the different helper T cell subsets. So I think that that's an important thing to see here. However, this figure shows more helper T cell subsets than we are going to talk about in class today. In fact, the ones that we're actually going to be talking about at all in this class are the ones that are shown in your textbook, which are the helper T cell subsets that are shown here. You will likely want to use figure 8.14 in your studying, as this is a really nice overview of the helper T cell subsets with the list of cytokines that induce differentiation, the transcription factor, and the cytokines that are produced, as well as the function of these cells. For the most part, I am going to stick with talking about the exact cytokines that are on this slide. And so this is really the view of these cytokines that you want to be thinking about. There are a few different ways that some people think about some of these cytokines. Different sources will give you slightly different cytokines for some of these cell subsets. But this table is really kind of going to be the master table that we look at for all of this information. When helper T cell subsets were first really described and first thought about, we didn't originally know that there were all of these subsets, or even all of the subsets that were on that previous slide, which are some of the ones we really think about. At the beginning, we only knew about two major types of helper T cells. These two major types of helper T cells were known as Th1 cells and Th2 cells. Um, and so the idea for each was that they sort of almost opposed each other. That these were the only choices. We now know that there are more choices that our helper T cell can have, but the big original T cell subsets were Th1 and Th2. Um, so here is some information about the Th1 cell. With Th1 cells, our activating cytokines are cytokines that induce differentiation. In particular, are IL-12. And so when I think about Th1 cells getting polarized, when I think about this Th0 cell becoming a Th1 cell, I really think about IL-12 as our main cytokine. When that Th0 cell receives IL-12, that makes the cell start making a new transcription factor known as TBAT. And you can see TBAT here. Once our cell starts to make TBAT, that pushes the cell towards making a particular cytokine, which is interferon gamma. You will see slightly different situations. You'll see on this slide, it also says TNF. On another slide, it also says IL-2. When I think Th1 cell, I think interferon gamma. I do not want you to think about IL-2 specifically as a Th1 cytokine. You should think about it as a general T cell cytokine. When we remember, we did see interferon gamma being produced by CD8 T cells as well. But in this case, we are seeing it from our CD4 type of cell, the Th1. And when that interferon gamma is produced, it can have a few different types of activities. One of them certainly is helping those CD8 T cells, those helper T cells. But the biggest thing that we see with interferon gamma coming from our Th1 cell is that these Th1 cells are now able to activate macrophages. So our CD4 T cell is going to produce interferon gamma. That interferon gamma is going to bind to the interferon gamma receptor that's on macrophages. And our macrophage that has previously phagocytosed something, you can see some bacteria that are in the phagosome of this macrophage. That macrophage, when activated, will get better at killing things that have been phagocytosed or bacteria that are living internally. And so you can see an activated macrophage here. A lot of the things like reactive oxygen species and other parts of macrophage killing are going to be more efficient and expressed at higher levels. And so this is a really good way that we can turn on these macrophages to kill anything they may have phagocytosed. In some cases, even to kill things that they may have phagocytosed that may be partially inhibiting that response. And so we often see Th1 cells as being really key for activating macrophages. What you might imagine here is that our Th1 cell is going to be particularly good at helping us eliminate things that live inside phagosomes of macrophages. Since the point of the interferon gamma made by the Th1 cell is to activate macrophages to kill things in the phagosome. And so typically when we think about Th1 cells, we think about protection against intracellular pathogens. So some kind of pathogen that's within a cell, either something that has been phagocytosed, that's inside of a macrophage, where we want to get that macrophage active to kill whatever's inside the cell, or something that's inside a cell that needs to be killed by a CD8 T cell. So we're killing some kind of intracellular pathogen with a Th1 cell. You can imagine that the IL-12 in the first place was made following some kind of PRR signaling from one of those intracellular pathogens. And Th1 cells can be involved in a number of different disease states as well. We also have the one of the first characteristic described helper T cell subsets, which is the Th2 subset. Th2 cells, again, are polarized when our initial Th0 naive T cell receives a particular cytokine from its innate immune cell during activation. In this case, that cytokine is IL-4. And the figure I showed you from your textbook earlier talks about IL-4 here. When that naive T cell receives IL-4, it will start making its characteristic transcription factor, which is called GATA3. When that cell turns on GATA3, it will start to make a number of different characteristic cytokines. These include IL-4 and IL-5. As it says here, they also include IL-13. Your textbook doesn't say that, but it's actually pretty straightforwardly also there. And so this one is pretty simple in terms of this figure compared to the one from your textbook. One thing that is a little bit confusing to people sometimes about Th2 cells is that Th2 cells both are polarized by IL-4 and make IL-4. There's some argument that there may be some other cells and cytokines involved in polarization. Some people talk a little bit about a cytokine called IL-33, but we're not going to deal with that right now. When we see the production of the Th2 cytokines, IL-4, IL-5, etc., we are going to see a different set of responses. So now we're not going to have activation of macrophages. Instead, these cells are going to do a number of different things. One big thing that we're going to see is some production of IgE from B cells in order to induce mast cell degranulation. We also will see some IL-5 that can induce eosinophil degranulation, and we can see these cytokines also involved in mucus secretion and peristalsis. And oftentimes, when we think about Th2 cells, this mast cell activation is particularly key. Remember that mast cells all have these Fc receptors on their surface. Specifically, they are Fc receptor epsilon receptors, which means that they bind to the Fc portion of the IgE antibody. So your mast cells are all coated with IgE bound to their Fc receptors. And if antigen comes along, it can bring two different Fc receptors with their bound IgE close together, as you can see here. This is called cross-linking, and that can lead to a signal in the mast cell, leading the mast cell to degranulate. Basically, again, it's just secreting its vesicles full of stuff. This includes things like histamine and other types of inflammatory mediators that hopefully will be able to kill this extracellular antigen. And so here you can see we're not phagocytosing. Instead, we're trying to kill that extracellular antigen. And usually when we think about Th2 cells, we often think about things like helminths, which are a type of parasitic worm that may be too big for our cell to be able to phagocytose. So it is an extracellular pathogen, not an intracellular pathogen. And the best way to kill that pathogen is going to be to secrete toxic stuff at it so that it can die, or try to do things like push it through your GI tract really fast to get it out of your body ASAP. Th2 cells, as we'll see more when we talk about allergies, also have a big role in allergies. I often shake my fist at my Th2 cells because they do all sorts of wrong things. And so while our Th1 cell was really important in activating, in helping us get rid of intracellular pathogens, Th2 cells are particularly important in helping us get rid of extracellular pathogens, particularly parasites like helminths or other eukaryotic parasites. So here you can see sort of the overall original view of what we understood about helper T cell subsets when we just thought about Th1 versus Th2 cells. So you can see that we can either have this Th1 type response that's shown at the top, where our antigen presenting cell makes IL-12 when it activates the naive T cell. That IL-12 turns on the production of the transcription factor TBAT in this helper T cell that pushes this cell to make or to become a Th1 cell and make interferon gamma. And this will be particularly important for helping us deal with intracellular pathogens. Alternatively, if our antigen presenting cell has some PRR get triggered that induces it to make IL-4, that will make our naive T cell turn on GATA3, the transcription factor, and that will push the cell to differentiate into a Th2 cell and make cytokines like IL-4, 5, and 13. And this will be particularly good at helping us combat extracellular parasites like worms or other kinds of eukaryotic parasites. So we were going to activate macrophages with the Th1 cell, while we're going to activate mast cells with the Th2 cell. As was also described pretty early in our understanding of helper T cell subsets, the particular cytokines that are made by different helper T cell subsets can also change the way different B cells will do class switching. So if we make interferon gamma, remember that interferon gamma comes from Th1 cells, that is going to help make B cells do class switch to things like IgG3 and IgG2A. Those are really good at helping deal with some aspects of particular types of pathogen responses, where we're going to do things like inhibit IgE production, inhibit IgM, inhibit IgG1. Alternatively, if we are seeing IL-4 and IL-5, we're going to get a different profile of cytokines made by the B cells in the periphery. Now we are going to see IgG1 and IgE, as well as perhaps IgA. So the way that our helper T cells are activated will also influence the responses that are made by our B cells, so that we'll get class switching to different isotypes of antibodies. There's one other thing that was understood in our initial understanding of helper T cell subsets. And that is the idea that helper T cell subsets don't sort of act a little bit in isolation. So not only are our antigen presenting cells producing some cytokine that can push a Th0 cell towards being a Th1 or Th2, but those cytokines also provide some other feedback. And so when a cell gets IL-12, it does turn on TBAT. That does turn on interferon gamma and promotes the cell becoming a Th1 cell. At the same time, this inhibits, so here you can see the inhibiting symbol, this inhibits GATA3. So not only do we push the cell towards becoming a Th1 cell, we have also inhibited that cell from becoming a Th2 cell. We've sort of had a little bit of a feedback loop to push all of the sort of immune responses that are happening right now towards Th1. Not only does TBAT turn on interferon gamma, it inhibits IL-4 and IL-5. And so once we start making a Th1 response, in response to a particular antigen or in a particular situation, that Th1 response further amplifies itself, as well as inhibiting any Th2 responses that may be going on. The same thing happens with a Th2 response. IL-4 both turns on GATA3 as well as inhibits TBAT. GATA3 turns on IL-4 and IL-5 to give us the Th2 phenotype. However, GATA3 also inhibits interferon gamma. So we can see that when we start to make a Th2 response, we get positive feedback towards having more Th2 response and inhibiting a Th1 response. And so we sort of get our immune responses pushed towards a general Th2 type of mode. We see this actually in different mouse strains, that they tend to be either Th1 or Th2 biased. As a person who has a lot of allergies, I know my immune system is very Th2 biased, so I tend towards more Th2 responses. And these two types of cells have a lot of feedback. This is, again, going to be something that's really important when we talk about allergies. So for a long time, we just had Th1 and Th2 cells. And we have since described a number of different helper T cell subsets. The next subset that I want to tell you about is one that at first you might find frustrating with its name. And that's because you might say, oh my gosh, when we did complement, we saw that immunologists were bad at counting. The C proteins in the classical pathway, they didn't go in order. Immunologists really don't know how to count. And now you're going to look at this T cell subset and say, oh my gosh, they went Th1, Th2, and then to this number. They really don't know how to count. But actually, the naming here is quite helpful, and I think that you'll end up liking this name. Because the next T cell subset that I'm going to tell you about is actually not the next one described. Going out of order here a little bit. But the next one that I want to tell you about is a cell known as a Th17 cell. And so Th17 cells are a very important helper T cell subset. They were described in the early 2000s. And Th17 cells are made when our antigen presenting cell sees some kind of pattern recognition receptor that induces the production of the cytokines TGF beta, IL-6, and IL-23. Your textbook lists all three. I want you to be aware that for a Th17 cell, you need a combination of cytokines. So for a Th17 cell, you have to have TGF beta, but you also have to have another cytokine, most famously IL-6, though of course this figure in your textbook also lists IL-23. So if you were listing for me Th17 cytokines, you have to have TGF beta plus one other cytokine, like IL-6 or IL-23. You can't just have TGF beta. Hopefully you'll see why as we talk about some things later. IL-23 is a cytokine that is closely related to IL-12, which was the cytokine that polarized Th1 cells. And in fact, because of some similarities between IL-23 and IL-12, it took scientists a while to actually describe Th17 cells as distinct from Th1. And I think that there's still a couple places in the literature where things get a little bit murky between the two. When you look at these cytokines that are polarizing the Th17 cell, one big thing that you want to note is that one of those cytokines I've listed here is IL-6. IL-6 is a cytokine that you have seen earlier in the semester. IL-6 is one of the inflammatory cytokines that we saw that was turned on when we saw an inflammatory response. And so some of you are actually measuring it in the lab. So you can think about IL-6 as being sort of real serious inflammation. And so with a Th17 cell, the key thing is we have this cytokine TGF beta plus inflammation. That's why I said you have to make sure that you are specifying the two cytokines with Th17s. You need TGF beta plus inflammation we can go with either IL-6 or IL-23 here. When the naive cell gets these cytokines, it will start making the transcription factor ROR gamma T, which you can see here. And when that cell starts making ROR gamma T, it will become a Th17 cell. And the Th17 cell is defined as not only making ROR gamma T, but making a particular cytokine, and the cytokine is IL-17. So that's actually how we named these. The Th17 subset is that they make IL-17. So the numbering actually does make a little bit of sense. You can see that Th17 cells also do make IL-22. And we have learned that the Th17 cells particularly are really important for activating neutrophils. Often in barrier tissues. And they're really important for kind of protection against fungal and extracellular bacterial infections. So our Th17 cells are big for neutrophil activation. They also can contribute to a number of different disease states in terms of autoimmune and inflammatory diseases. Another important helper T cell subset, also described in sort of the first decade of the 2000s, is known as the TFH, or T follicular helper cell. So the T follicular helper cell doesn't have a number in its name, so it's not helpfully named like the Th17s by which cytokine it produces. However, it is named helpfully based on where exactly it acts to do its job. So our T follicular helper cell, again, is generated when our Th0 cell is polarized by particular polarizing cytokines. In this case, the polarizing cytokines are a combination of IL-6 and IL-21. When our Th0 naive T cell receives IL-6 and IL-21, that induces that cell to start making a transcription factor called BCL6. Once our cell starts making BCL6, that pushes it to become a T follicular helper cell. And this T follicular helper cell is now going to make the characteristic cytokine IL-21. And IL-21 is very important for activating B cells and helping with an antibody response. B cells particularly receive help in a part of the lymph node known as, or a part of the secondary lymphoid organ known as the follicle. And so these are the T cells that are providing help to B cells in the follicle. So TFH stands for T follicular helper. And this is also particularly important in a place called germinal centers. So here you can see our T follicular helper cells that are polarized when the Th0 cell receives IL-6 and IL-21 to make the transcription factor BCL6 and then produce the cytokine IL-21 in order to help B cells in the follicle. We have one other important T helper cell subset that I need to mention to you. These T cells, this T cell subset is extremely important. It's a very, very big deal. It is also one that has had a controversial history in immunology. Because in fact this T cell subset was described, and then because of some experimental technical issues, it was largely abandoned by the field, to the point where people were like, what? I published a paper on what? No, I don't believe those cells exist. Those cells don't exist. What are you talking about? Those aren't real cells. In later years, these cells were sort of rediscovered, and now we know about their importance, but their name got changed. And so there is another name. I will use the other word, but just like with danger, sometimes when I use this other word, I feel a little bit like um, I'm going to be struck by lightning. And this is going to lead to one place where I want to highlight kind of a weird phrasing that immunologists like to use. So this final group of helper T cell subsets that I want to tell you about are a group of T cells known as regulatory T cells. They are sometimes referred to as Tregs. The old name for Tregs that now, of course, no serious person would ever say that they ever believed in, was suppressor T cells. And so the word suppressor or suppression in immunology is a little bit of a dirty word. In fact, as this figure shows, these regulatory T cells act to turn off other T cells. And so you can see that here it's shown as suppressing autoreactive T cells, but really it's just sort of a way of turning them off. You can see that these cells are listed as inhibiting inflammation and inhibiting other responses. In general, the word suppression is a word that immunologists don't like to use. When you hear immunologists talk about regulation or regulatory T cell, generally when we say regulation, we mean turn off because we're so skittish about using the word suppression. So these are the regulatory T cells. They do regulation. And generally, when I say regulation about T cells, I am saying that I'm saying off. It's like I'm saying suppression in code because I can't say the word suppression without getting struck by lightning. So you will notice that I talked about regulatory T cells a little bit differently in terms of how I've presented them to you. As you will see moving forward, there are two ways that regulatory T cells can be induced. One of those two ways looks very similar to the subset polarizations that we have seen with the other types of subsets that you see here. And that type of Treg generation is described on this slide. But what I want you to remember is this is only one of the two ways we can generate Tregs. Tregs are generated in this way when our naive T cell, our Th0 cell, receives a cytokine. And in this case, this cytokine is just TGF beta. Remember that in the case of IL-17, our cell was receiving TGF beta plus an inflammatory cytokine like IL-6 or IL-23. When the cell receives just TGF beta, that's a sign to shut things down and turn things down. There isn't that inflammation coming along as well. And so if the cell sees just TGF beta as opposed to TGF beta in combination with the inflammatory cytokine, that cell is going to become a regulatory T cell. When that cell gets TGF beta alone to become a regulatory T cell, it turns on a transcription factor known as FOXP3. And FOXP3 induces that regulatory T cell to make cytokines IL-10 and TGF beta. And IL-10 and TGF beta turn off other types of responses. And so this is going to inhibit all of our other types of immune responses. You might imagine regulatory T cells are pretty important for things like protecting us from autoimmunity, and are super important in a lot of different immune responses. As I mentioned, there are two different ways we can make Tregs. In both cases, the cell ends up making FOXP3. So all Tregs, no matter how they got turned into Tregs, are making FOXP3. All Tregs, no matter how they got turned into Tregs, are making IL-10 and TGF beta. And they're always inhibiting other types of responses. The difference is how the cell got told to turn on FOXP3 and how the cell started down the Treg path towards the Treg life. The answer is that some Tregs are actually induced in the thymus. So for every other subset of cell that we're talking about in terms of our helper T cells, the T cell comes out of the thymus as a Th0 cell and doesn't decide which helper T cell subset it's going to become until it sees antigen and gets some cytokines from an APC in the secondary lymphoid organ, like the lymph node. But some Tregs become Tregs in the thymus and stay Tregs for life. And so you can see the Treg coming out of the thymus here and having it still had to upregulate FOXP3. So we're still seeing a FOXP3 dependent process here. This Treg that came out of the thymus will still make IL-10 and TGF beta in order to inhibit other cells' responses. And this production of Tregs from the thymus is actually the fourth outcome that we see for helper T cells the fourth outcome that we see for selection of T cells in the thymus. So if you remember, we saw the situation where cells got low or no signal, and those cells failed to be positively selected. They kind of went around, they were useless, they could not bind to MHC plus peptide. They were hoping for signal, and then they died because they never got any signal to tell them they did a good job. So those cells will die by neglect. We had those cells that got sort of the low intermediate signal and were thus positively selected. So they got enough of a signal to be told, hey, yeah, you're good, you should survive. We want to keep you around. You should become a T cell. So those are the positively selected cells that survive. We saw the cells that had the very high signal. And remember, those cells were autoreactive. Those cells were actively deleted or negatively selected because they had the ability to be autoreactive. And then I told you that there was this other thing that happened with the cells that had a sort of high medium or high intermediate affinity. These cells are getting closer to the danger zone. Remember that we have this danger issue in terms of all of our T cells have to be a bit self-reactive. These are on the higher side of self-reactivity. They're not so high that we want to kill them. They could be useful, but we don't really want them to leave the thymus in a way where they can be dangerous. And so these cells will actually become Tregs in the thymus, and they will be able to regulate other cells that may be autoreactive. So we take advantage of these cells' higher level of self-reactivity to help us combat everybody else's self-reactivity. So you can see these cells are coming out of the thymus as Tregs because they had sort of a little bit of a higher affinity for antigen, and they will go on to be Tregs. And so Tregs are able to suppress or inhibit other cell types in a bunch of different ways. One of those ways is that they can inhibit the actions of many other cells due to making TGF beta and IL-10, and those directly turn off things like CD8 T cells, other T cells. So we're inhibiting cytotoxicity, we're inhibiting other T cells, we may even inhibit antigen-presenting cells. The regulatory T cells also have huge amounts of IL-2 receptor, particularly IL-2 receptor alpha. This was actually discovered even before FOXP3, and so their IL-2 receptor alpha levels used to be what defined them. And they can act by basically soaking up all of the IL-2 to deprive other cells of IL-2. So regulatory T cells can both inhibit other cells by active mechanisms through TGF beta and IL-10. Sometimes they do it by touching other T cells. Sometimes they don't. They can also do it by more passive mechanisms of cytokine deprivation. As we have learned about all of these different T cell subsets, many of the same principles that we first understood about Th1 versus Th2 cells stayed in place. So in every case, we still had some polarizing cytokine that turned the cell, at least in the periphery. Regulatory T cells that are made in the thymus are a little bit different, but regulatory T cells made in the periphery are like the other helper T cell subsets. We have some sort of polarizing cytokine that polarizes our Th0 cell into our type of cell type. We have some master regulator transcription factor that's made. We have some cytokine that is our characteristic effector cytokine. But what we also have seen is that all of our different helper T cell subsets have feedback with one another. Not only do they induce positive feedback to induce more of their type of subset, but they also will induce negative feedback so that they inhibit the differentiation of other cells. And so you can see, for example, a Th2 cell will lead to more Th2 cells. It will inhibit Th1 cells. It will also inhibit Th17 cells. You can see Th1s will inhibit Th2s and Th17s. Tregs will inhibit both. And so we can see feedback between all of these different subset types, where each subset reinforces differentiation towards its own type as well as inhibiting differentiation towards the others. And this summary shows you the details of all of the polarization towards these different types of helper T cell subsets. So in all cases, we started with a Th0 cell. That Th0 cell received some kind of cytokine that induced differentiation. That pushed the cell type towards a particular polarization towards one of these 5 subsets. And that subset was in fact defined because the cell makes a particular transcription factor. Once the cell makes its special transcription factor, then we will see that cell making its characteristic cytokine, as is listed here. And that characteristic cytokine allows the helper T cell to do its function. And so what you can see is really if we were trying to say something that all T cells do, it's activate or it's interact with other cells and influence their responses. And that's really the only way we can describe what's going on by all of these T cells because their changes are so broad. In the case of helper T cells, the key is that they are also all doing this process by producing cytokines. And so we think of our helper T cell or our CD4 T cells as our big cytokine producers. On Friday, we're going to really spend most of our time thinking about B cell responses in the periphery. We just finished our discussion of T cell responses in the periphery. However, I do have a few things that are sort of on the border between the T cell and the B cell responses that I want to talk about for the rest of our time today. So, as I said, we're moving into thinking about B cell responses in the periphery on Friday. Our T cell responses in the periphery largely started in the secondary lymphoid organ, particularly in the T cell zone. The B cell responses in the periphery are also happening in the secondary lymphoid organ, but now they're happening in a different area, often the follicle, which is the B cell zone. And so we're going to be in a similar location. When we think about how we activate B cells in the periphery, there are a couple of different ways that we can activate B cells in the periphery. These are known as either T-dependent or T-independent B cell activation. Some antigens are thought of as either being T-dependent antigens or T-independent antigens. Really, the big difference between T-dependent and T-independent antigens has to do with how exactly the B cell is getting its signal to, because B cells need signal to just like T cells do. And in the case of a T-dependent antigen, the B cell is getting signal 2 from a T cell. It's getting that signal 2 in the form of CD40 on the B cell, interacting with CD40 ligand on a helper T cell. In the case of T independent antigens, signal 2 is coming in another way. Because I'm really talking right now about T cell responses in the periphery, I am specifically pointing out a couple things about T dependent responses. And T dependent B cell responses are kind of the main B cell responses people like to think about. And so when we think about T dependent responses, one thing to realize is that they are only going to be responses to proteins. If we're thinking about polysaccharides or lipids or nucleic acids, other types of antigens, often those are T independent antigens, though we will eventually see some more info about that. And with T dependent activation, we see some important things happening. So with T dependent activation, that's when we see class switch, that's when we see affinity maturation, that's when we see immune memory. While we can activate a B cell in a T independent manner, we don't really see those same processes. And so T dependent B cell responses are those responses that are going to allow us to have class switch, that are really going to give us this good proliferation of the B cell, that are going to give us good memory cells, and that are going to give us that higher level of affinity for our antigen. So in order to make a T-dependent response, our B cell needs help in the lymph node or in the other secondary lymphoid organ. And oh my gosh, that's happening from a CD4 T cell, the cells that we were talking about a little bit earlier. And so you can see that B cell will receive signal from its antigen. However, that B cell is in fact a antigen presenting cell. Remember when we talked about MHC class 2 presentation that I told you that B cells are able to internalize the antigen using receptor mediated endocytosis and present that antigen on MHC class 2. That means, of course, that they will be presenting to a helper T cell, and that helper T cell can produce some cytokines as well as produce CD40 ligand to interact with the B cell's CD40 in order to give that B cell both a signal 2 through CD40 and sort of a signal 3 with those cytokines. Specifically, this process is happening in the follicle of our secondary lymphoid organ, like the lymph node, and will eventually lead to an area called the germinal center that we'll think about on Friday. All of this action is done by one of these helper T cell subsets that we talked about, the T follicular helper cell. So this is sort of a way that really thinking about B cells is a part of or related to thinking about helper T cell subsets. And this is the reason why we had to leave off our B cells after we finished B cell development, because we couldn't actually learn more about some of these details of what happened to our B cell to make B cell responses until we knew about T follicular helper cells. So you can see our T follicular helper cell leading to B cell activation here. So what you will notice is that our B cell receptor will be binding to antigen on that B cell before to sort of start this process of B cell activation. And you can see that our B cell will then do receptor mediated endocytosis to bring in that antigen on into the cell. That antigen will get degraded and presented on MHC class 2, specifically to a T follicular helper cell, since those happen to be the cells in the follicle that are happily specialized to produce cytokines and to help B cells. And now that T follicular helper cell will both produce CD40 ligand to activate CD40 on the B cell, and that T follicular helper cell will also produce cytokines. As you saw earlier, that T follicular helper cell is likely producing the cytokine IL-21 in order to help that B cell to fully activate that B cell. One other thing to note about this is that The T cell in this case is a T follicular helper cell. The T cell in this case is not a naive T cell or a Th0 cell. In order for a cell to be a T follicular helper cell, that cell must have previously seen antigen to have been activated. And so in order for our B cell to be properly activated, our T cell also has to be properly activated first. So our B cell activation can't happen until we've had T cell activation and until we've had our T cell be polarized to a T follicular helper cell. This also is really important in thinking about some aspects of B cell responses. If you recall, I told you that T dependent responses tend to happen only with protein antigens. This really has to do with the details of T follicular helper cells activating B cells. Remember that in order for our B cell to fully be activated, it needs to get help from a T follicular helper cell. T cell, and that involves the B cell presenting its antigen on MHC class 2. T cells only can recognize peptides on MHC class 2. MHC class 2 can't present things other than peptides. And so we're really only going to see peptides or proteins that are turning on our T follicular helper cell to allow our B cell to get activated. We can also think about the fact that our antigens that we see from our B cell and our T cell don't actually have to be identical. And so I want you to look closely here. Our antigen in this case, you can see has a red part and a blue part. You could imagine those as perhaps being two different epitopes. This is a very low resolution image. And you can see that the B cell receptor binds to the blue part, the blue epitope. That leads to internalization of that antigen, and that leads to that antigen being degraded for MHC class 2 presentation. And so you can see the degradation leading to some peptides from the blue part, some peptides from the red part, etc. The T cell is going to recognize some part of that was presented on MHC class 2. In this case, it is the red part, not the same epitope that was recognized by the antibody or by the B cell receptor. It's a different epitope. However, because of that presentation, this B cell will get help. And when this B cell produces antibody, it will produce antibodies that bind to the blue epitope, not the matching red epitope that the T cell saw. This becomes really important in a process called linked recognition. We're going to see linked recognition again on Friday, but I know many students find linked recognition a little bit confusing, so I want to talk through it once now. Again, we'll see it again on Friday, and we'll talk through some details of a problem in class on Friday. So here you can see, don't worry about where it talks about polysaccharide and toxoid, we'll deal with that later, but you can see a B cell that is binding to an antigen with multiple parts. You can think of them as multiple epitopes, the blue flower part and the red stick part. You can see that the B cell internalizes that antigen and that antigen is degraded inside of the lysosome, and some part of it gets presented on MHC class 2. And that could be a different part, like the red stick part, which was different and was what the B cell was binding. Now we're going to turn on our T follicular helper cell. Now we're going to interact with our T follicular helper cell. Remember that our T follicular helper cell already had to be activated. It needed signal 1 and it needed signal 2. So that's going to be a whole other thing. We have to make sure we have good T cell activation to make a good B cell response. Our T follicular helper is going to provide CD40 ligand to bind to CD40. It's going to provide cytokines. And now our B cell is going to start to make antibodies And those antibodies are going to bind to the initial antigen that our B cell receptor had, because that's the only one it has, thanks to allelic exclusion. And it's now going to make a lot of antibodies to bind to its antigen, in this case, the blue flower. This is also going to be something that we can take advantage of if we want to make antibodies in the lab by making certain combinations known as hapten carrier conjugates. And we'll talk about those more on Friday. So, like I said, on Friday, we're going to be talking about peripheral B cell responses, and we'll see a little bit more of linked recognition again. Thank you so much for putting up with the recorded class today. And I look forward to seeing you all on Zoom on Friday. Thanks.